pleasure to introduce the next speaker, who is Helen Mayberg. She received her MD from the University of Southern California and trained in neurology at the Neurological Institute of New York at Columbia, and did a postdoctoral fellowship in nuclear medicine at Johns Hopkins. She is presently a professor of psychiatry, neurology, and radiology, and the Fuqua Chair in Psychiatry, Imaging and Therapeutics at Emory University. She spearheads a multidisciplinary research program which has been defining a neurology of depression, and she has longstanding interest in neural network models of mood regulation in both health and disease. We're very privileged to be able to hear from Professor Mayberg this afternoon.
Thank you for the invitation. I always feel apologetic when I see genes to cells to knock out animals and engineering at this level, but I think that to study human disease, behavioral disorders, that starting from the patient and trying to slowly move in has been a strategy that hopefully can now leverage the kind of tools that Inscopic has and what everyone is here to kind of discuss. So I want to be both provocative, but to share kind of some of the trouble that happens when you try to model a complex human disease and what things you can do and what things you can't do. This is mostly grant supported and not NIH supported. It's really been through foundation support that a lot of the work has been done. And I'll be talking about off-label uses of both unapproved and approved investigational devices toward these goals. I also want to just introduce my team up at the front. And again, it requires this interdisciplinary effort of surgeons and clinicians and engineers and modelers and basic scientists and imaging scientists to actually pull this together. So I think the bigger concept outside, and we've heard about it through a lot of these animal models, is that we're assuming and using terms like circuitopathy, that these disorders can be modeled in this way, and that what we need to know is what is an essential node that we might do something to that will impact the disorder. And that, again, I'm going to take the point of view from one of the questions posed this morning by Tom Insel, was what's a circuit? And I'm going to argue from the point of view that there is an anatomical structure that has function within it, and that we're coming from that perspective down. And if you are going to manipulate a circuit with a tool like deep brain stimulation, which is what I'm going to talk about, that you're going to need to know kind of, A, what is it that you want to do? What symptom, what do you want to treat? And I think when you look at Parkinson's, there's both primary symptoms that you target and secondary symptoms that you don't. In some ways, it's because you want to avoid side effects from treatments you already have versus maybe you want to stop the progression or the development of resistance. You need to know what it is or want to know what it is you want to have happen, both to know that you've engaged a target, that you can know what's happening over time, what's the endpoint you're aiming at. You need to know where you're going to stimulate, and is it a critical node? Is it to define a circuit to go outside the circuit? You need to fix something that's broken, need to go beyond what's broken. And then, I mean, fundamentally, who are you going to do it to? And is there the notion that for a given disorder, one size fits all? And just like in pharma, failed trials are about assuming one size fits all, and nobody gets 100% market share. And when you're targeting circuits, that maybe we ought to build that into our model. I think that the other thing about thinking about focal modulation with large devices is that the catalyst for even approaching this over the last 20 years, and many things haven't changed much over the last 20 years, but what has changed is the advances in stereotactic neurosurgery and the tools surgeons have to be precise in an operating room. The availability of evolving devices that allow what kind of implants, what kind of recording devices we have, and then the availability of imaging tools that gives us precision. And I think having the circuit models both described in animals but really characterized in humans as imprecise as low resolution as they are, is a starting point. And that has gotten us to a point that there are current applications of invasive neuromodulation. Tim Dennison talked about them this morning, that can be used to alleviate suffering in people with advanced Parkinson's, essential tremor, dystonia, and experimentally in the neuropsychiatric realm, and I'm going to talk about that in depression. So again, conceptually, to get into what needs to happen to get to this point, you have signs and symptoms, as in Parkinson's, because that really is the model that we've all followed. You have pathology. You heard this exquisite talk this morning with the direct and indirect and medium spiny neuron manipulations. We have the pathology. We have the circuit. But it was actually the ablative neurosurgery techniques that actually honed us in. And we heard the description of Ben Abid kind of using high-frequency ablation, reversible ablation, to test hypotheses that were already there about the circuit that was well known. And then the details are worked out in the patients. But to appreciate that motor symptoms like rigidity and tremor are affected, but not freezing or falling, and not cognition, and not autonomic instability, and not mood dysregulation. A circuit disorder is a number of different pathways and circuits, and you kind of pick where you're looking and where you're modeling. And I want to throw that out there now just because I think that's part of the discussion point for later in the work groups. In depression, we really try to take the same approach. There's a laundry list of signs and symptoms that make up the syndrome. It's very, very similar to Parkinson's: mood, motor, cognitive, autonomic, just with a different balance and an emphasis on mood. There's not pathology at the same level of nigral degeneration, but you can induce focal lesions in the frontal lobe, produce depression. You can induce lesions in the basal ganglia in certain places and induce depression. You can have degenerative disorders. You can look at atrophy and see abnormalities in the hippocampus, in the ventral medial frontal cortex. And one can start to have putative nodes that one can organize into putative nodes in a circuit, and one can start to test that with functional imaging and say, what do depressed people have that non-depressed people don't, and start to pick up candidate regions. And through that process, and then also a causal model, how does a metabolic signal, and these are FDG PET scans, how do they change as you go from sick to well? And one can start to see which regions are best behaved and start to, again, hone in on what regions might you care about in more detail. Can build a model. And I think this became a really critical issue of where to go next if you're going to start thinking about putting an electrode in a patient's brain who has a major mental illness. What thing do you want to fix? And to me, the feature of clinical depression, the psychic tension, inability to move, the mood part, when that's gone, the rest you can kind of do rehabilitation on. So when you can't make that feature move, one has to actually consider that which symptom do you want to go away first. They may not all change at the same timeline. So even experimentally, we can build an experimental probe to say where is the negative part. And it was through looking at the overlap between mood induction with activation in the ventral cingulate and turning off of prefrontal cortex, two regions that seem to move in a yoked manner, that was the opposite pattern that you saw when people went over time from sick to well. That the question was, can you tune this system like Parkinson's using those putative targets? And, it seems like ancient history now, but that was the logic. This relationship among many could be approximated with laughable precision. This was called find the blob of psychic tension that turns down with depression recovery. Ask a surgeon to make a guesstimate and put a probe in it. Turn it on and see what happens. And what happens is you can start with overactive, Brodmann area 25. You can turn it down and affect changes both locally and remotely. And at the clinical level, what happens? A rapid change in depression ratings over the first week, a regression when you turn it off for clinical recovery after a battery implant, and a change with chronic treatment over time that over 2 months starts to separate people that don't get better from people who do. As you repeat that, just to actually kind of get a feel for the time activity curve of high frequency stimulation in this region, you actually see that if you go from 6 patients to 20 patients, go from 6 months to a year, repeat it with a different cohort, different surgeons, in this population of patients, there is a change in previously intractable patients that go from sick to well and stay well with continued stimulation. So to me, that puts us, after many years, into a place to say, what are we really doing to this brain region? What is happening in the network broadly? We have some target areas to try to study and hopefully model with non-human models. Again, what I like to think of as a reverse translation to then have a forward translation, so a bidirectional translational relationship among different levels of resolution. So at the clinical level, it's been 10 years. And it wasn't just Area 25, of which there have been at least 8 publications of small samples with anywhere from 40 to 60% response rate sustained over months to years. There have been studies in the reward system, in the ventral capsule, in the nucleus accumbens, in the medial forebrain bundle. And the big problem has been that ramping it up to clinical trials has been a disaster. The ventral capsule study by Medtronic stopped after 30, active didn't breach sham. The St. Jude Medical Broaden study, results still not out, did 90 subjects, halted for futility after half the subjects enrolled. And so we have a problem about, does it work, does it not work, should we stay in this or not? We've got the Brain Initiative, DARPA, everybody wants more devices, more fancy, put it in these people, and the question is, dead stop, what are we maybe doing wrong? And so, you know, in trying to think about this as in small experiments, in a single lab, one seems to have predictability over time, but ramped up, it seems to spin wildly out of control. What is it that we're doing wrong? And I think we go back to the basics. We need to know where, we need to know who, and we need to know what we want to have happen. And maybe we need to kind of try to work out the sources of variance in the human to figure out what thing in the animal that we might want to model. And so this I just want to show as the illustration of the tactic we took, which is kind of starting to unpack. Trust that if people get better, it's real. Not assume that it's some magical process that just is maintained over 10 years. But to actually try to think about the where first, because that's the easiest to fix or modify. And it turns out that the actual location of the electrode can't explain responder, non-responder, except when one looks at the function. So the function of the network from the PET scan change, we never actually miss changing the activity in the target. What we miss is whether or not we affect change remotely in the distributed network. And it isn't just absence of an effect in one place. It's absence of an effect quite remotely in fairly predictable areas. And if one spends any time thinking about connectomes with diffusion tractography, you can look at this image here and actually imagine the cingulum bundle, the frontal projections of the uncinate fasciculus as actually not having metabolic changes over time with chronic stimulation. So one can model what is present about people who get better or people who don't as a way to kind of refine what's going on with when it's working, with when it's not. And by modeling very grossly the volume of tissue activated, applying it to the tractography maps to understand what pathways am I affecting when we effectively change behavior, and ask what do people who recover share. They share targeting the cingulate bundle, paths to the ventral medial frontal along the uncinate fasciculus, and pathways to the accumbens and thalamus subcortically. And that if you look at non-responders, they're in the complementary color, they partially get better, but they didn't actually have impact in the tractography map on the full circuit. And if you look at how patients just naturally evolved between 6 months and 2 years who did better, and model it purely based on the tractography map of what you impacted, one can see that the conversion of non-responder to responder can be explained by actually moving into the full circuit. So again, circuit used in a very gross manner. And so how do we then have a causal or prospective experiment to prove it? Well, we now have our connectome surgery target rather than an area 25 target. We can drive around on a single subject tractography map until we find the location that is the intersection of these 4 critical bundles. We can implant there, stimulate there, never change it over 6 months, and we go from a 40% response rate to a 73% response rate with much less work for the team and no other changes made. So maybe this is generalizable to thinking about all of these connectomic targets, whether it's in the medial forebrain bundle, in the ventral capsule, they're all white matter targets. What is the connectome that one is impacting? Can one model it? Can one use the clinic to actually understand that? So the second point is, well, if one can explain the variance of where, how about the variance of who? And this is a problem that has plagued everyone in terms of the no one gets 100% market share. In depression, the numbers are that first treatment, 40% will achieve remission with the first treatment that they're offered. That over time, it's trial and error to get people to the treatment that's best for them. And that's not getting into the realm of these ECT-resistant patients that are the focus of the brain stimulation experiment. But by having even these gross, low-resolution human maps that can be articulated with imaging, which only gets better and better, we can know that certain aspects of these systems change differently when people recover from therapy versus drug. But we can also start to identify that people's resting state at baseline within the same circuit can be in different configural states when they will or will not respond to therapy or drug. So just as an example with PET imaging, a low insular state will predict that you will do great with therapy and terrible on drug. But the same region in a high activity state at baseline will predict that you do great on drug and therapy won't touch you. And if you actually take the people who can't respond to anything compared to the people that can respond to either of the other treatment, they have high activity in area 25. We can get similar kinds of maps when we do it with resting state fMRI, and it's led us to test the hypothesis that what's wrong with the targeted DBS network, and it turns out that the pathway in the cingulum bundle is abnormal with PET, with white matter, and with its functional connectivity to the area 25 target. The more abnormal this mid-cingulate state is, the sicker you are, and that it turns out that this pathway is totally normal in people who get better on more conventional treatments. So we suddenly have an area that we might want to model. Thinking back to Corey's comment today about diseases of the body relate to the brain, this area actually modulates the autonomic nervous system. This is where you get changes in autonomic tone. It projects in Peter Strick's latest work to the adrenal medulla, that we start to have points of intersection of where willed action, autonomic function, all of these central features of why you can't move when you have pain might be regulated. So to just try to figure out, the last issue is the what, the trajectory of recovery. And I think this is most important for thinking about how to model these kinds of complex diseases, how to think about compensatory strategies, that what we've learned from stimulating in area 25 in these patients is immediately in the operating room, when you are in the convergence of those four pathways, you can evoke a reset out of this kind of local minimum stuck state. Patients say all kinds of crazy things, but what they do notice about themselves over time is there's a rapid shift in state, but then there's a chronic recovery period that requires ongoing stimulation and rehabilitation. And thinking about this, it's not a linear going from sick to well, but there may be stages that we need to model these systems differently. We know at the PET scan level that changes locally happen rapidly and stay over time, and even when you turn it off after 6 months, you don't get a big rebound. But if you look at the remote areas, you don't get changes with acute stim. It requires ongoing stimulation, and you turn it off, and that's where the rebound is. So thinking even about Dr. Feng's comment this morning about development, that certain pathways may actually have different developmental trajectories, may have different plasticity curves, that I think we have to think about this going forward. And we also need to think about how can we use tools to actually measure those different opportunities. We can start to look at the reset with electrophysiology in the OR, just as they do in Parkinson's. The left hemisphere does not change like the right hemisphere, but they certainly work together. We have to have models that will actually look at the electrophysiology signal on both sides at the same time, but not expecting them to be matched. And now we have tools like Tim described this morning, where we can make monitoring of brain signals chronically, like telemetry. And with the brain radio, we can actually measure the physiological signal off of area 25 in real time, multiple times a day, and actually look, now that we know who the right patients are, where to put it, and have a predictable clinical recovery, how is the signal changing over time, and just take the opportunity to measure it and see, and start to develop other fun measures beyond patient self-report, whether it's actigraphy or heart rate variability or changes in facial expression that we can monitor and use as regressors with our brain signal. We've got all ways to refine what we want to do, and I think for what would be useful models, and I put this in to just use the last minute, is that make us a better connectome. You've got optical control, but you can see we don't want just one pathway. We've got to be able to modulate one pathway and then manipulate another, all at the same time and with different kinds of speeds. Maybe chemical control is just as good. And obviously the network monitoring, like within Vista, is going to be key. But I've got to have an explanation. Do I need cell-specific tuning? High-frequency stimulation doesn't do a bad job, but maybe we could do better if we could refine it. But the question is where? Which pathways are mediating the reset versus the plasticity? Build an animal model of treatment-resistant depression. An animal model that responds to fluoxetine is not good enough. We don't need that. We need to break a system multiple times. And work like Huda Akil is doing is the kind of thing is, how do you make systems that actually match the human condition that we need modeled? The last thing I want to end with is back to the patients, because I think the patients have to be part of this discussion, and I think we have representation of advocacy groups there. If you have a condition in which you cannot escape, and it doesn't necessarily kill you as a degenerative process, you have to actually, we need to figure out how we change the culture so that our partnerships kind of include the patients in terms of what is their risk tolerance as well when we start to have clues that we're going in the right direction. So it is about always managing a benefit-risk ratio. But again, watching what will happen in industry after failed trials, we have a real problem, even though in the interim, while they don't even publish their data, we actually have revised and figured out how to fix things that they never considered. We've got to have a way to work back and forth to recover things when the first pass by industry goes wrong. Thank you.
Kip Lodewig from the Mayo. So a lot of the assumption in operating at a node in the circuit, especially with DBS, is that DBS is either essentially exciting or inhibiting the node in the circuit. But there's some really interesting work out of, say, Matt Johnson's lab showing that because it's non-naturally activating, it may inhibit the soma, but still stimulate motor efferents and actually create a temporal time course where, because of the circuit it's connected to, inhibit for the first 3 milliseconds past a pulse, then excite, et cetera. How do we need to get to that level of granularity to understand these systems? And in kind of the vein of the summit, what are the technologies you see? Is LFPs enough to get to that sort of precision?
So I think that, I'm very simple-minded about this. I'm trying to become less linear in the thinking experimentally because I've got to deal with the patient. I think that the first question is measure the LFP locally, hedge your bet, and measure remotely with EEG, and actually see if the signal tracks with the behavior in any kind of predictable way. I think once one has a signal, of which there is an emerging signal both locally and remotely, one can look at their interrelationships, their coherences like we heard before, and then start to break the system by selective models that say, I'm going to hold one part of the circuit and one change that I've identified stable, and I'll look to see what other part of the remote system I can turn off and see. So I think that the step we're at now is to actually just kind of see where the next step should be. And again, if that's to be accused of being too pedestrian, then I'll take the criticism. But I think, a company that has a device where you measure off the lead and can get a readout, it's of the whole system. The question is whether or not it has high fidelity enough or it avoids the noise to actually see if it's usable. But at the same time, not to do 100 people to realize that was a bad idea. Use small samples, get in fast, have a high bar that you've got to get a good readout, and then at the same time collect the other data so that you can revise. So I think even in the patients, we've got to optimize the opportunity to iterate particularly when we know we're getting a good signal clinically. Then we are reverse engineering what we're doing. We're not altering at their expense that we do something that isn't in their best interest.
Helen, this is Goping Feng from MIT. I just have a quick question. Have you ever tried to do fMRI in the patients under anesthesia, whether this CG25 overactive is persistent or is just temporal? So,
again, the safety issue and actually the artifact issue of doing repeat fMRI with the device in is a problem. Mayo, they do fMRI in the scanner, but not with the device implanted. At Cleveland Clinic, they've done some studies with the device implanted. There's terrible artifact around the lead. So if you need to know what's happening locally, even as a seed, to look at its relationship to the rest of the network, you'll get a change because you have an artifact. Same with the white matter change. There's abnormal white matter. The question is do you turn on oligos with stimulation and actually induce plastic changes in this network at different rates? And I think that now the technology and the safety issues to either devise leads that and devices that can be in the scanner safely without artifact, and to do it, but it's actually a technical limitation right now. And I think that's where, if one buys the findings and can construct models that look at certain of these pathways, one could set it up in animals and test it directly in the animal models and know.